Thank you.
So if there are no questions, we will move on with our last topic for today, investigating ascariosis and West Nile disease through mathematical modeling to aid public health interventions. That would be Elisa Fesce will present from the University of Milan. So go ahead, Elisa.
Yes, thanks. Let me try to share my screen. Okay, hopefully you can see my presentation, right?
Yes.
Okay, so thanks for presentation. Thanks for allowing me to be here. So I will briefly show you the work I performed in my PhD about the use of mathematical modeling to investigate infectious diseases. So as I told, we are talking about infectious diseases, in particular, diseases that are communicable, zoonotic, and emerging or re-emerging. And the tool we are using to investigate them is mathematical modeling. Why mathematical modeling and what are mathematical modeling? So any kind of use of mathematical rules and formula to describe a phenomena can be considered as a mathematical model. So also kind of progression or any kind of rule. In particular, then we use models that are compartmental models and are based on a system of differential equations. What does compartmental models mean? It means that we have a population and we just subdivide it in different compartments, so in different subgroups of the population that have specific characteristics like being susceptible for an infection or being infectious or infective or just being recovered, like in the most famous SIR model. And then the model we will use are based on a system of differential equations. So a system means that just that all equations are contemporary true. And differential equations are equations that can show us the dynamics of a phenomena, and in particular represents the rate of change, the change of the number of something, in our case of individuals, on time, on any kind of variables, but we are most interested in the variation on time usually. The strength of mathematical model is that they can help us in investigating mechanisms underlying this variation of numbers. So as coming back to our aims, we are talking about mathematical modeling applied on infectious diseases. The main important thing that this kind of model can help us do is understanding the spread of an infectious disease, because as we saw, they are a dynamics representation of the processes and can help us in understanding those mechanisms underlying this spread. And these two characteristics can help us in a better understanding of the spread, but also in understanding the best way to control infectious diseases, and can help us in predicting the future spread of these infections. So mathematical model allows us to perform in silico simulations, so we don't need to test actively the thing we want to test, but just we can perform it by running simulation on a laptop or a computer, and it gives us information that are a priori. So we just can simulate and see what will happen. And the most important thing is that they can give us the possibility to simulate hypothetical scenarios. So like, for example, answering to what if questions, like suggesting what would happen if we can just apply a control strategy that is not applied yet. That can help us in save time and resources before, but also investigating a disease. These characteristics are very important, of course, if you are speaking about emerging diseases for which we rarely have a previous solid experience to rely on. But also if you are talking about zoonotic diseases that are that usually involve several species and for which it's very hard to assess field experiment, and especially if they also involve wildlife. So why the choice of analyzing emerging and zoonotic diseases? Of course, because their incidence is increasing in the last years, and especially the incidence of zoonotic diseases, but also because they have complex cycles, they involve several species, they're new, and for all these reasons is often the amount of data that we can have is often limited. That leaves hard the understanding, predicting, and avoiding and managing of these diseases. So our aim was to investigate this mechanism underlying zoonotic emerging infections and investigate the best way to reduce human infection risk. The two diseases we focused on were West Nile virus and Parasitology. Both of them are emerging or re-emerging diseases that can cause severe outcomes in humans and are both wildlife-borne. West Nile virus involves mosquitoes and birds, whereas Parasitology involves wildlife-borne, involves raccoons. Both of them are present in Italy. The first is endemic, the second one is just introduced, can be introduced. And for both of them, we have several knowledge gaps till now. So my work was aimed at answering 4 main questions. The first was how to improve our current comprehension of West Nile virus spread. The second one was to try to investigate which are the bird species that are mainly involved in West Nile virus spread. The third one was how to reduce the human infection risk. As the fourth question, we try to understand which is the best intervention strategy to apply to avoid the risk of having Parasitology on a territory. So for answering the first question, so how to improve our comprehension of West Nile virus spread, we first validated a mathematical model based on a system of 13 differential equations representing birds and mosquitoes. We validated it in order to explore the dynamics of the disease within birds and mosquitoes in Lombardy region. And we use it at first to estimate a kind of outbreak probability in each month, finding that it's very likely to have an outbreak of West Nile virus, especially in June and July, suggesting and highlighting the importance of the surveillance and further investigation about this disease. Then we investigate, investigate how the temperature can affect West Nile virus spread, finding that temperatures lower than 14 degrees do not allow for West Nile virus spread. Third, that was our current aim, we performed a sensitivity analysis on epidemiological parameters. But first of all, what the sensitivity analysis is and why it is important to perform it. As we told, mathematical models allow us to investigate mechanisms of spread. Those mechanisms are represented by parameters included in the model, but not all parameters has the same importance in the output of the model. So there are some parameters that cause a small variation in model estimate when varying them, and there are others that can give us a very big variation in model simulations, meaning that there are some mechanisms of action that have a high weight on disease dynamics, while others doesn't. So while investigating which parameter has the major effect on model simulation, we can better understand which are the mechanisms that have the biggest importance in driving the disease spread. So we investigated different epidemiological parameters, and we estimate how does a change in those epidemiological parameter estimate influences the West Nile virus prevalence in mosquitoes. The parameters we investigated were birds' recovery rate, bird competence, bird susceptibility, and mosquito biting rate, finding that birds' recovery rate and mosquito biting rate has the greatest effect on model estimates, whereas competence and susceptibility has a lower effect. Then, trying to answer the second question, that was which bird species are mainly involved in West Nile virus spread, we mainly use the same model, but instead of changing epidemiological parameters, we then changed the avian species included in the model. We changed the demographic parameters of the species involved in order to see if differences in demographic parameters can affect West Nile virus prevalence in mosquitoes. And we found that mainly is the abundance of birds that affects West Nile virus prevalence, and it's a negative effect, so that the less birds we have, the higher West Nile virus prevalence we have in mosquitoes. The third work was centered on control strategies against West Nile virus. So we just include in model simulation 6 different intervention strategies that were elimination of overwintering mosquitoes, larvicide treatment, adulticide treatment, the reduction of mosquitoes breeding site, and 2 interventions on birds population that were birds removal and the removal of birds breeding sites. All intervention strategies were tested at 3 different intensity, 20, 50, and 80%. And we found that the intervention against the mosquito population has a better effect compared with the one against the bird population. In particular, was the reduction of mosquito breeding site having the best results. In the graph, we can see on the x-axis, the time, and on the y-axis, we can see the reduction of the risk. So the highest curves are the ones that show the best results in risk reduction. So reduction of mosquito breeding site was the best one, followed by elimination of eggs and larvae, and followed by the elimination of adult mosquitoes. The elimination of overwintering mosquitoes show a good response at the beginning of the season, whereas the effect go losing efficacy during during time. Whereas the intervention against birds show an opposite trend, so they are likely to improve our infection risk. So as fourth answer, we try to understand again which was the best intervention strategy, but this time to reduce the risk of infection by Lysascaris prochonis, and Ascaris. We then built another mathematical model based on a system of 3 differential equations representing the host, represented by raccoons, the parasite, and the free living stage, represented by parasites' eggs. And then we introduced in the model 3 different intervention strategies. The first was aimed at the culling of raccoons. The second one was represented an anthelmintic treatment, and the third one, feces removal. That means the removal of free living stage. And we focused our work on 2 main questions. So the first one was, can the intervention eliminate the number of eggs? And the second one was, how long does it take to eliminate eggs from the environment? We found that host removal has the best results, followed by anthelmintic treatment, and then feces removal. This work, of course, show us the important role of the different population involved in infection spread. On the one hand, showing us how different characteristics of hosts and vectors, in our case, but mainly of host species, can affect infection spread. And on the other hand, tells us the importance of identifying the population that is needed to be targeted to have an efficient control strategy. So that's all. Thanks for the attention, and thanks also of Università degli Studi di Milano for the possibility, but also Fondazione Edmund Macken and Università di Trento that gave us the mathematical support for the developing of mathematical models, but also Regione Lombardia and l'Istituto Profilattico Sperimentale della Lombardia e dell'Emilia Romagna for sharing their data with us. Thank you. Maybe you have some question.
Yes, thank you. Thank you very much for your interesting presentations. There are questions for Elisa? If not, I have one, Elisa.
I think there's also a question in the chat, but just go ahead.
Okay. Okay, I will expose my question, then we can look on the chat.
Sure.
How much we can rely on the mathematical models in terms of, you know, prediction, forecasting? I mean, there are a lot of mathematical models now that are going on a lot of parasites, a lot of species. So in terms of, you know, because you have this expertise, how much we can rely?
I think it depends on which mathematical model are you talking about. Mainly it depends on the assumptions and the data we have about the disease. I mean, also if it's a predictive model, we need to rely on some data. So it depends on the amount of data we have and how do we use them. So it's, as for a field experiment, it depends on the assumption and how is it done. I mean, I cannot tell you that they are reliable or they are not. In our case, I think we should need a little more data to have valid predictions, because we only use 3 years of data. So about West Nile virus, we had a good fit of models to data, but 3 years are not very
enough.
Yeah, they're enough for having, for example, for our sensitivity analysis was enough, because it's a kind of, we're not trying to predict the future. We are just looking at the effect of different parameters. So 3 years was enough. But if you want to really predict what will happen next year, we need stronger assumptions to base, I mean, as our starting point. But it mainly depends on what kind of model are you looking at. It's a quite hard question to answer.
Anyway, thank you very much, Elisa.
Thank you for the question.
So we have here
Serena,
from
I think.
Serena, yeah, Baliscaris has been reported in Europe or in Italy.
Okay.
She's asking.
At the time we performed our analysis, we didn't found any Baliscaris in Lombardy raccoons, but I think now they found maybe some in Tuscany, but I'm not sure data are available yet. So I think we need to wait for a publication about that.
Thank you, Elisa. There are other questions or maybe comments?
They asked from your group.
Yeah, from your group.
What from my group? I mean,
probably
if we found, yeah, but if we found, no, it's not. I mean, probably some of our group will be involved, but I currently don't know, because I'm working on the mathematical part. So yeah, probably you have to ask to Nicola.
Okay, right.
No, they are not for my. May I speak a bit?
Yes, you can.
Yeah, sorry. No, it's not from our group, but these positive have been found in Foreste Casentinesi Park. Colleagues with which whom we collaborate, but is independent research. And about Europe is very common in Germany. 60% of raccoon of German raccoons are positive, so it's fairly common.
Grazie Nicola, ciao.
Ciao, Serena.
Okay, so I see that she was satisfied by the answer. There are any comments or questions? Because we are in the end of this first session. Any? Okay, then. I must say that I have to thank you, all of you, for sharing your afternoon and your topic interest. And it was great that were various topics. And now, because we know each other, perhaps we can collaborate as well. We have our email address, so it was a start to know each other and the interest of each other. So thank you very much once again. Many thanks as well to Alice Vismara, our invited speaker, and see you next week on Thursday, 4 November, with our second edition of Spring Parasitology. Thank you very much.